Let's understand key drivers of CRISPR market. One definitely is increasing prevalence of genetic diseases as there is more awareness and more diagnosis and availability of more molecular diagnostic techniques. High number of cases of rare genetic disorders are being screened and more prevalence of genetic diseases. It was estimated that 1 in 25 children are affected by genetic condition. So, with increasing prevalence of genetic disorders coupled with rising demand for gene therapies, more and more number of gene therapies and biological being approved by regulatory authorities across US, Europe. So, increasing prevalence and coupled with increasing penetration or high rising demand for gene therapy are key drivers for this CRISPR market. Next. Also, rising demand for precision medicine instead of using a common drug, precision medicine such as CRISPR, which help in elevating the disease condition for one specific person based on his genetic condition or his genetic mutation. That is a key driver for CRISPR market, and also supportive regulatory landscape as one therapeutic drug from a company called CASGEVY has already been uh, uh, or for a drug called CASGEVY has already been approved for sickle cell disease. And there is a clear regulatory motivation for other CRISPR therapeutic based companies to gain regulatory approvals in near future. I'm moving on to drivers for CRISPR in agriculture and diagnostics. So in agriculture as well, uh, companies are partnering and companies are developing or using CRISPR technology to uh, to improve agricultural productivity, to develop many disease-resistant crops or other nutrient crops. Of course, as of now, in agricultural productivity, companies are more focusing on smaller pilot studies and on smaller crops, tomato and other master. Still, companies are doing research to apply the CRISPR technology on row crops or large scale mechanized to apply on a mechanized basis on large scale crops. However, definitely many companies, big companies are using CRISPR technology, are partnering with CRISPR technology based companies to apply this technology into to increase the agricultural productivity. And in diagnostics as well, in diagnostics already there are marketed products using CRISPR technology to detect COVID or respiratory diseases, HIV, and other diseases. And because of COVID, a few companies such as Sherlock and Mammoth Sciences have got emergency use faster regulatory diagnostic approvals, and now uh, such products are already in the market and are being used for testing various diseases. So there is high penetration of CRISPR-Cas technology in diagnostics because the technology is very specific, highly sensitive, easy to use, less turnaround time, and hence these key parameters help in rapid penetration of CRISPR-Cas within a diagnostic space. Of course, big drivers there are market restraints. One is high cost of CRISPR-based therapies because of the complexity, because of the cost involved in development of CRISPR-Cas. For example, the marketed drug CASGEVY is cost around 2.2 million per patient, and such high cost drugs where there will be issues with the reimbursement and accessible the affordability of such high priced or such high cost drugs is in low income or low income countries. So that is one of the restraints. Second, ethical and regulatory considerations because CRISPR-Cas technology is itself modifying the genes, modifying the genome, inserting a new DNA sequence within the genome that is specific inside the human body. So in some cases, using human embryos to develop or to insert the gene. So these are ethical and regulatory considerations which acts as a hindrance to adoption of this technology. And last, potential off-target effects because of the complexity of the genome. Uh, Even though companies are researching a lot to develop high specific guide RNA, because the most important component within the CRISPR-Cas is guide RNA, which guides Cas9 to specific location within the genome. Even though the companies develop and many advanced techniques or AI-based computational techniques are available to design guide RNA, still the genome is very complex, and there are many off-target effects where guide RNA or plasmid can bind to some other location in the genome and cause some mutations within the genome, which we do not know until and unless it is infested, or it is infested into the human. So there are definitely potential off-target effects and safety concerns which are hindering the adoption of this technology.